Hi there, Andy Mackey here. My little video on coronavirus, understanding how it works. And it's related to the SARS virus, the severe acute respiratory syndrome virus. This is a second version of that, and the COVID-19 disease. Here's an electron micrograph of the virus particles, and a beautiful little thing. Here's a mock-up model of what the virus particle would look like. You'll see lots of different coloured proteins on the outside. So it's made up of a membrane which has lots of proteins within it. There is the spike glycoprotein, which is the thing that binds to human cells. There is also the transmembrane protein M, which you'll see crosses the membrane 3 times like a zigzag, and there's also a membrane protein E, yellow on this diagram as well. So a number of proteins around the side, and then inside is the nucleocapsid protein, which helps just bind together the genetic code, the RNA, in this case single-stranded RNA, which is the viral genetic code that it will multiply within human cells. So, unravel the RNA code. If we were to stretch it out into a long line, then we can understand that it's made up of different bits. So this is from Wuhan. It's got an open region frame 1A, 1B, and then code for the S protein, the E protein, and the M protein. And it uses the host cell's components to produce proteins that will replicate the code and produce the S, E, M, and N proteins. So here's the virus particles here being replicated. So the human cell membrane has a receptor on it, ACE2, and this is the thing that makes it very specific to humans and why it's jumped into the human population, because the virus, the spike glycoprotein, binds to this receptor on human cells. And as a result of that, it then makes use of the apparatus within the human cell. It's taken into the human cell, and then the genetic code, the RNA, is released into the human cell. And as a result of that, it then hijacks the cell to do all its work for it. So the RNA that we looked at a minute ago, with the different code for the different proteins, is then, as I say, hijacks the human cell apparatus. These are the ribosomes that read the code and convert the code into proteins, 2 little polypeptides, which then form a protein, and that protein is a polymerase, which just means that the RNA, the genetic code, can be multiplied. The viral code can be multiplied lots of times, and the rest of the RNA codes for the S, M, and E proteins. And again, it hijacks the human cell's apparatus, the ER and the Golgi, to produce those proteins. So you've got the spike protein in particular there, which then they all reassemble with the genetic code in the middle to produce new virus particles, which are then, as I say, multiplied many times and then released into go and infect lots of other cells. So you get the whole pathway there from it binding with receptor ACE2, releasing its genetic code, producing proteins and more. So you get lots and lots of virus particles. And the ACE2 receptor on human cells is very common in cells around the lungs in particular, as well as the heart, and hence the respiratory problems that it causes. Hope you found that helpful in understanding a bit of cell biology, a bit of molecular biology on coronavirus.